Hello everyone. In today's lecture we will talk about peptide mass fingerprinting PMF. There's a mass spectroscopy based technique which is used in proteomics laboratories to identify proteins. Just recall from the earlier lecture, in a proteomic studies we analyze thousands of proteins present in a cell, and it comprises of two steps. First is separation and second is identification. So all the proteins present in the proteome in a cell in a given condition, those all are separated using a two-dimensional gel electrophoresis, where you see thousands of spots, and each spot that comprised of one protein generally. And the second step is, if you want to identify these proteins, some protein of interest or all the proteins in fact, so you can cut these spots, and after cutting the spot you can treat with some protease like trypsin, and after hydrolyzing the protein into peptides, those peptides can be identified using peptide mass fingerprinting, or another MS based technique is tandem mass spectroscopy. And in this lecture we'll talk about peptide mass fingerprinting, the technique which is used very commonly for identification of hundreds of protein per day in a typical proteomic laboratory. We all know that beside peptide mass fingerprinting and tandem mass spectroscopy, there are two more techniques which are used to identify protein. One is Edmond degradation. That's a chemical sequencing method. This also we have discussed in earlier lecture. We use phenyl isothiocyanate, a chemical, in order to sequence peptides and proteins. And another method is Western blotting. Western blotting is used if you have thousands of proteins and you want to see whether a particular protein is present in the complex mixture. So you only need to have antibodies against that particular protein. You can, the two-dimensional gel or even SDS page, the gel can be transferred into some blotting membrane and then incubated with a particular antibody against that specific protein, and Western blotting is used to see whether a particular protein is present or not. But if these proteins are unknown, or you want to identify n number of proteins, in that case Western blotting is not done. Peptide mass fingerprinting was developed in early 1990s, and is also known as protein fingerprinting. It's an analytical technique, and where proteins are identified without sequencing. For example, Edmond degradation and tandem mass spectroscopy, proteins are sequenced first and later on they are identified based on the sequence. But in peptide mass fingerprinting there is no sequencing required, and that's also advantage of this technique. So, and it's very rapid. In today's time there are almost 500 proteins per day can be identified in a typical proteomics lab. And it's a high throughput protein identification technique because a large number of proteins are identified using PMF. And there are three steps in peptide mass fingerprinting. In first step, there's a formation of experimental spectra. The protein of interest is cleaved using a protease, some enzyme which can hydrolyze protein into peptides, and that mixture of peptide is run into mass spectrometer, and the experimental spectra is obtained. Then theoretical spectra is also obtained. Theoretical spectra means if genomic information of that organism is known where we are doing proteomic studies, the genome of that organism is converted into proteome, so we have all the protein, theoretically all the protein sequence of that organism available. Then each protein is cleaved with the same enzyme which was used to make experimental spectra, and theoretical spectra are made from the database. And in the third step, the experimental spectra obtained is matched with the theoretical spectra available from the database, and with whichever experimental spectra matches best with the theoretical one, that is the protein of your interest. So it's a very simple technique where theoretical and experimental spectra are matched in order to know the identity of a protein. So in the first step, for example, we have a two-dimensional gel electrophoresis, 2DG gel, and one protein we are interested in identification. So this protein, this spot from 2DG can be excised. It can be excised, and after excision means there will be single protein in one spot. We are assuming that. So in gel, digestion is performed. The protein is digested with say for example trypsin. Trypsin is very commonly used protease which hydrolyze lysine and arginine C-terminal in peptides and proteins. So after treatment with trypsin, you get n number of fragments depending on presence of number of lysine and arginine in the protein, and this mixture of peptide, when this is run on mass spectrometer, we get a large number of peak depending on number of peptides present. So this is called experimental spectra. So unknown protein of interest is cleaved into peptide, and these peptides they are used to produce experimental spectra in a mass spectrometer. And we know that from earlier lecture, MALDITOF or ESITOF, they are ESI, electrospray ionization, and matrix assisted laser disruption ionization. They are two very commonly used techniques which in proteomics in order to ionize peptides and proteins. In the second step, after obtaining experimental spectra, if genomic information are available, so the genomic information is converted, the genome sequence is converted into the proteome, and you have a database where proteome of that organism is available. And proteome means all the proteins present in the cell, in the genome. So then you use computational program. This is in silico. So use computational programs. There are algorithms available which convert each protein into peptide using the same enzyme which you have used in preparing experimental spectra. For example, use trypsin to make experimental spectra. So here also there are computational programs which will cleave each protein sequence present in the database with trypsin. So for example, if they are say there are four protein in the proteome, so you will get 4 different spectra each for a single protein. So you get depending on the number of protein present in the database, same number of theoretical spectra you have. So in this you use the same enzyme like trypsin, and theoretically you acquire the spectra. So this is all theoretical, because we know that in mass spectra you just observe the mass. So theoretically, when you cleave a protein into peptide, a sequence of protein into peptide using some protease, you know the precise mass of each and every peptide. So based on that precise mass you have obtained in silico, you can construct a theoretical spectra. That's what is done by computer here. So you get a number of theoretical spectra depending on proteins present in the proteome of that organism. So once you have both theoretical and experimental spectra available, experimental spectra is experimentally you have made using mass spectrometer, and theoretical in silico. So no, the third step is matching. So experimental spectra is matched with the theoretical spectra. So in the protein of interest is cleaved into peptide to form the experimental spectra. For example, this is experimental spectra, and the same time the proteome is cleaved. Cleaved by the same enzyme to get number of theoretical spectra. Now this experimental spectra is matched with all these theoretical spectra, and with whichever theoretical spectra it matches best, that is the protein of interest. And this matching is statistically analyzed. Statistically analyzed means how much is the matching. It should be the best out of all the theoretical spectra available, and one of the parameter of statistical analysis is sequence coverage. For example, this is the sequence of a protein, and here you have experimental peptide list. So you see that not all the peptides are matching with the theoretical spectra, theoretical sequence. Only the peptide which are shown in the red color, they are matching exactly, rest are not matching. So there are different reasons to that, we'll discuss after this. And this is called sequence coverage, means the number of amino acids which are matching between the theoretical and experimental spectra, that's form sequence coverage. So sequence coverage should be high. in order to have the best match. So in summary, you have a complex mixture of protein, that's a proteome. Proteome is run on 2DG, and this port you want to identify that is excised from the gel. It is proteolyzed using trypsin, for example. You get a mixture of peptide. Those peptides are run on a MALDITOF or ESITOF, and you get spectra that's called experimental spectra. Now, though simultaneously you have a protein database available for that organism, which proteome you are studying. So that proteomic database, in proteomic database, computational program will cleave each protein from the database into peptide fragment using the same enzyme which have used for preparation of the experimental spectra, and so you get number of theoretical spectra available. Now the experimental spectra is matched with the theoretical spectra in order to identify a protein. Whichever theoretical spectra matches best, that's the protein of your interest. So not all peaks between theoretical and experimental spectra, they are matched, and there are several reasons to that. For example, peptide length versus MS range. Mass spectrometer, they have a fixed range which peptide mass can be detected. Sometime if some peptide is beyond range of the estimation of MS mass spectrometer, in that case that peptide will not be part of the experimental spectra, hence it will not match, although it will be present in the theoretical one. But in experimental spectra it would be missing. Or if enzyme has missed a cut, or there is a non-specific cleavage. For example, you are using with trypsin, but some molecules are not cleaved. Some sites are not cleaved by trypsin because of different reasons. So in that case, that peptide will not be part of the theoretical experimental spectra, or there is an incorrect open reading frame. That also leads to different peptides, different peptide mass, and mismatching of both the spectra, or if they are amino acid substitutions. Because remember, in theoretical and experimental spectra, we are just matching the mass. If there is a, with any reason, if there is a difference in the masses, the matching will not be there. So another reason could be ion suppression efficiency. In mass spectroscopy, you ionize peptides. Some of the peptides, they are not ionized well, so they are not part of the experimental spectra. Although theoretically those peptides would be there in theoretical spectra, but experimentally that peptide is not there because it was not detected by mass spectrometer. So that's how it could be another reason of not matching both the spectra. Or in the experimental peptide list, if there is any kind of contaminant. Contaminant could be, for example, you are treating protein with trypsin, and trypsin is a protease. It can hydrolyze its own molecule also. One trypsin molecule can hydrolyze another trypsin molecule. So in the mixture of peptide of your unknown protein, there will be mixing of peptides coming from trypsin itself. So trypsin peptide also would be there, which naturally will not match with the theoretical spectra. Or if you have any other mixing of, say, hair or skin keratin from the user, so those keratin protein is present in the sample additionally, so that also will give rise to peptide fragment when you treat with trypsin. So those will not be matching, of course. Or in ionization, if you are using, say, MALDITOF, in case of MALDI, matrix molecules can form cluster, and those clusters also, they give some peak, and those peaks are part of the experimental spectra. Those are not in theoretical one, they are coming from only matrix. Or there is any unknown contaminant. If there is any contaminant, you cannot, that contaminant will not match. Similarly, very common one is post-translational modification, any kind of modification which has occurred to the protein after translation. So that will not be part of the theoretical spectra, although it will be there in experimental spectra. For example, post-translation modification in case of eukaryotic proteins. There are modifications like there is acetylation. If there is acetylation, it will add 42 Dalton mass to the peptide where this acetylation is present. So mass will be extra in case of experimental spectra. Similarly, if there is a phosphorylation, phosphate group is attached to serine or threonine side chain. So in that case, 79 additional mass is present to that peptide in experimental spectra, although it will not be there in theoretical one. So that's how that peptide will not match. Or there is a glycosylation. Glycosylation, it varies in mass, 162 Dalton to 3000 Dalton mass. So if there is any kind of change in the amino acid, that is leading to difference in mass of the peptide, that will lead to mismatching of both the spectra. Methionines are oxidized. Methionines can be oxidized to methionine sulfoxide, for example. One oxygen is added, and that leads to 16 Dalton additional mass. Similarly, methionine sulfone, they are also formed after oxidation, where two oxygen groups are added to, oxygen atoms are added to sulfur, and that leads to 32 Dalton additional mass to that methionine, and same time to the peptide where methionine is present. Or even there are chemical modification to cysteine. Carbodomethylation of cysteine can occur, and that leads to difference of mass of 57 Dalton. Or if there is any amino acid deletion in the protein. In theoretical spectra, deletion is not there, but actually when protein has matured and active, in that case, if there is some deletion present, that there will be difference of masses in that peptide where deletion is present between theoretical spectra and experimental spectra. Or they are splice variant. Splice variant is also another very common phenomena which leads to difference in the protein sequence. If protein sequences are different, so naturally there will be mismatching of the spectra from the theoretical and experimental values. So, but one can take care of these problems to some extent. For example, calibration can be done. Mass spectrometer can be calibrated to acquire the maximum number of peptides being produced. Or internal calibration is also possible, because if they are, for example, trypsin autodigestion peaks are present, we know trypsin sequence, we know where trypsin cleaves. So we all know if you hydrolyze trypsin with trypsin, what kind of molecular weight, what kind of peptide would be present. So those peptides can be omitted from the experimental spectra. They are made in background. So that's how matching can be improved. Or similarly, keratin protein is also known. We know the keratin fragment being produced after trypsin digestion. Those can be removed. To some extent, post-translational modification also can be added. For example, if there are two peptides which are not matching between theoretical and experimental spectra, and their mass difference is coming 79 Dalton. So we can assume there may be a phosphorylation, the one phosphate group may be attached to the peptide. That's why it is giving 79 Dalton mass additional. So that's how calibration is also possible. When you see result, you see that, for example, in this one, protein is giving match of the creatine kinase. And what additional information is available? Some score is 681. This score is basically cumulative number of considering the sequence coverage and the matching. So that should be the maximum in order to have the best match. And same time you have theoretical mass is given. Theoretical mass is the mass from protein sequence, and theoretical pi, this is a calculated pi from the sequence. Similarly, you have observed mass, that's 43 kilodalton, and observed pi. This pi and mass is coming from 2DGE. We remember from the earlier lecture, in two-dimensional gel electrophoresis, you can find out mass and pi. So you see the observed mass, observed pi, theoretical mass and theoretical pi. If you look at their similarity, you find that only the mass is very similar. The theoretical mass and observed mass is very similar, whereas the theoretical pi 5.34 and observed pi 6.2 to 6.27. They are drastically different. That's because mass doesn't change much. Only the pi can drastically change in between theoretical and experimental values, that we have also discussed in some earlier lecture. The reason of differences in theoretical pi and the experimental pi. And the sequence coverage, for example, here is shown 46%. It means out of the total number of amino acid, 46% amino acid are matched between experimental and theoretical spectra. The advantage of peptide mass fingerprinting is that only the masses of the peptides have to be known. There is no requirement of sequence. You don't have to sequence the peptide in order to identify protein. Only mass need to be known. Disadvantage is that the protein sequence has to be present in the database. The genomic information, the proteome database should be available for that organism which you are studying the proteomics in order to identify it. And another could be most PMF algorithms, they assume the peptide come from single protein. If there is a mixing of proteins in the protein of your interest, then in fact not only PMF but even Edmond degradation or other methods of identification, they are not valid. They cannot be used in order to identify the protein if there is a mixing